In the name of Allah, the most beneficent and the most merciful. Assalamu alaikum wa rahmatullahi wa barakatuhu. I welcome you in Pakistan International School Taif's virtual learning class for first year biology. Dear students, how are you all? I hope you are doing well at home. Dear students, we have started chapter number 2 before, in which we are studying biological molecules. We have started the topic carbohydrates. Today we will continue that topic and we'll see the third type of carbohydrates, that is polysaccharides. So let's get started. If you see this in this image, as we have discussed it before, that we have seen monosaccharides, oligosaccharides, and polysaccharides. So today we will discuss this type of carbohydrate, that is polysaccharide, and in that you can see there are two types, homopolysaccharides and heteropolysaccharides, and in that there are different types. What are these and how their structures are, properties, and all the stuff we will see in this topic. Okay, we will see polysaccharides. So in polysaccharides, poly means more, saccharides means carbohydrates, in which the long molecule in which many carbohydrates units are combined, are making the bond and by a condensation process, that is polysaccharide. In polysaccharide, it is the largest group of carbohydrates, and that is of two types. One is homopolysaccharide, in which homo means same, poly means many, saccharides means carbohydrate units. So in which lots of carbohydrates units, but which are all same units. So for example, in starch, glycogen, cellulose, and chitin we'll discuss here in this topic as well, about their structure as well. So then you can see that in these polysaccharides all the units are same, and that is glucose. So next one is heteropolysaccharides. Hetero means different, so in which all the monosaccharide units, they can be different. So they are agar, pectin, and peptidoglycan. They are heteropolysaccharides. So polysaccharides, actually they are very good energy reserves for all living organisms. If we talk about starch, that is plant carbohydrates, and glycogen, animal polysaccharides. So they are very good energy resources, as well as they perform the structural function as well. So examples are cellulose and chitin. We will see also their structure and function. Then we will see, first of all, about detailed structure and function of starch. Okay, starch is what? That is homopolysaccharide. It means all the saccharide units or monosaccharide units are glucose, which are present in that. They are formed by condensation process of hundreds of what? Alpha glucoses. So here we have discussed before that there are two types of glucose, alpha and beta. That is due to the position of a hydroxyl group at carbon number 1 in glucose. If that is below the ring, so that means that is alpha. It is above the ring, so that means it is beta glucose. So in starch, there are alpha glucoses which are arranged by glycosidic bonds. Starch is mainly present as a stored energy reserve in the roots, stems, and seeds, like vegetables, which are like beetroot and like carrots, radish, and stems also, which are edible, and seeds also like grains. Cereal grains, potato tubers are rich in source of starch, and if we just see about its chemical reaction, so starch is digested in oral cavity, as we know, by oral by amylase, and um, that is also digested in small intestine completely by intestinal amylase as well. The enzyme is amylase. In oral cavity, salivary amylase, and in small intestine, intestinal amylase that digest carbohydrates. On hydrolysis, it yields maltose. We have discussed before that maltose is disaccharide, and that is um, is formed by the hydrolysis of this starch and glycogen as well, and it is formed by two glucose molecules. It yields into two glucose molecules. So long chain of starch is broken down into maltose by maltase enzyme, and then it yields, then it is easier to just break it down into glucose by amylase enzyme. So the presence of starch is confirmed by what? By iodine test. So iodine gives blue color with the starch. It gives blue color with starch, and like if you put a few drops of iodine on cut potato, so that after some time it turns slightly bluish in color, so it means starch is present over there. So there are two types of starches, amylose and amylopectin. What are these? Actually, you can see there's not too much different, but structural slight difference is in the bonding. If we see, this is amylose, and amylose, this is what? Glucose ring. Okay, alpha glucose is there, that's why this is alpha 1 and 4 glycosidic linkage we have discussed in the previous lecture as well. So it linked with oxygen, and water is removed via condensation process. So lots of glucose molecules are bonded together by 1,4 alpha acidic bonding or linkage between to all the glucose molecules. So this is amylose, and these are long chains are formed. Just if we talk about amylopectin, amylopectin is having the same linkage, but it is in branched form. Amylose in chain form, amylopectin is in branch form. And if we just see this one, it's branched anywhere in oxygen. Then it is 1 and 6 linkage. 1,6 linkage means one carbon of one glucose atom and sixth carbon of other glucose atom. The sixth carbon is out of the ring. So this forms the chain structure, and from here the next chain is arised, and from anywhere the next chain can also be formed. So in amylose, one alpha 1,4 glycosidic linkage is formed, but in amylopectin, alpha 1,6 glycosidic linkage is also present. That's why it is insoluble in water due to the strong linkages. And in amylose, that is unbranched, a linear chain of glucose in which glucoses are attached. Okay, so if we just see the next polysaccharide, homopolysaccharide, this is glycogen, and that is also called animal's starch, and that is present in the liver and in the muscles as well. In the liver and in the muscles, that is homopolysaccharide and alpha glucoses are present, and they form the chains like this. And its sample can be confirmed by iodine test also, but it gives a red color. Why this gives a red color? Due to these linkages. Okay, so what happened here? Here is 1,4 linkage, but with the chain here is 1,6 linkage also. The structure of glycogen resembles with amylopectin starch, but glycogen is much more branching than amylopectin. So amylopectin is having less branching, but this glycogen is having like this beaded like structure. It is having lots of and very small units. They are having lots of branching system, branching structure as well. So now we will talk about cellulose. Cellulose, as you know, that it is the most abundant carbohydrate on the earth, and it is also a homopolysaccharide, and that unlike starch and glycogen, it is having condensation process, and hundreds of beta glucoses are condensed with one another. In starch, in glycogen, so we have discussed about they just form with alpha glucose the linkages, 1,4 linkages and 1,6 linkages, but here beta glucoses are forming the linkages. Due to this structure, like orientation or beta glucose's presence, it forms the structural carbohydrate of the plants and major constituent of plant cell wall as well. We know that cell wall is too hard. It gives a rigidity, it gives a strength to the plants to just get upright in the outer atmosphere. And cotton and paper, they are also pure forms of cellulose. So cotton is completely made up of cellulose. So cellulose having no color means it does not show any color to iodine solution. If we just talk about its structure, it resembles with amylose. But what is the difference here? Because amylose is unbranched, so this is also unbranched. But the difference is it is having beta glucoses. Due to this slight orientation, like slight change in the position of hydroxyl group, it is having the structure which is very hard and tough. How this is made? If you see this one, uh, if you see this structure, as I'll show you here, yes, this one, you can see each beta glucose molecule must be inverted at 180 degree from the previous molecule. 180 degree, how does it happen? We'll see here. This is one glucose molecule, okay, and this is the other one. You can see here hydroxyl group is above the ring, above in the ring on both of the sides. So this is 1, 2, 3, 4, 5, and 6. So on number 1 here and on number 1 here, hydroxyl groups are there. So how they will join? They are forming 1 to 4 linkage, but here the hydroxyl group is down. So this is harder to, this does not allow any type of linkage here. This is harder to link with both of them. They will make the bond with oxygen, and H2O will be removed. This is like condensation process. But how does it happen? To make this comfortable linkage, what will happen? You can see here, this number 1 molecule, this is number 2. If we just see number 1 and number 2 molecule, so this number 2nd molecule, this is oriented or inverted into 180-degree, means from down to up. So hydroxyl group comes along this hydroxyl group. So when they come along like this, so then this water will be removed and oxygen will make bond between these two molecules. So like this, when the other molecule will be combined, condensed with this one, so what will happen? This will orient, having like the 180-degree orientation, or 180-degree, we can say it will be inverted. So this will form, one molecule will be like this and the other molecule will be like this, then one like this and the other will be like this. So this will form linear structure, but it is having an alternate molecule having the orientation. So you can see here, this one, this group is here, so this one is down. If you see oxygen atom, one is here in the ring and other is down, like it's flipped downward. So this is very, very important, and then this makes it stronger. And when this changes, you can see here. So when you see this diagram, you can see here, there is 1,4-glycosidic linkage here, and now they will form a bond like this, flipped bonds, and you can see this orientation in all the chains. So these are the chain, not change, actually the linear chain structure, but not the branched one. So they are arranged upright and inverted manner. So then they form chains like this, linear chains, and then they form among them hydrogen bonding, and they form the crisscross arrangement. And this crisscross arrangement is very, very important for the strengthening of the cellulose. So cellulose is that polysaccharides which cannot be digested by human beings, but it is very, very important in the food, because it gives a roughage, like it makes our stomach full, and it just prevents the abnormal absorption of the food, like the people does not cannot have constipation or diarrhea due to accumulation of like vegetables and fruits, because their skins of fruits and vegetables are having cellulose in it. But herbivore plants, they are having symbiotic bacteria, those are which secrete cellulase enzyme, and this cellulase enzyme break down this long molecule of cellulose into disaccharide that is called cellubiose, and then this is further digested into glucoses. So if we talk about the next one, that is chitin. Chitin is as similar as glucose, and this is the second most abundant organic molecule on the earth. because it is present in the cell walls of fungi. We know fungi is a microorganism. Sorry, fungi is like it is um, uh, that is also unicellular but multicellular as well. Like we are having here mushroom. So mushroom is also fungi. So in the cell wall of the mushrooms as well as other fungi, it is having um, cell wall which is made up of chitin, as well as it is present in the exoskeleton of arthropods like insects. Arthropods are insects, and like beetle and cockroaches, etc. They are having exoskeleton, means which the skeleton which is present up on on the body, not inside the body like us. Because with the exoskeleton, arthropods can protect themselves from harsh environment, and well as they just save them from dehydration of the body, or dehydration of the body. So due to occurrence of chitin, a fungal cell wall is known as fungal cellulose, and it is the derivative of N-acetyl glucose amine, which is the modified form of glucose. So here we can say its structure, all the structure is same, the linkage actually is same, 1,4, but it is beta glucose here, and the linkages you can see here is upside and here is downside. But at carbon number, this is carbon number 1, this is carbon number 2, another molecule is present that is called N-acetyl group. This is called acetylamine, or that is called N-acetyl group, and means nitrogen, and nitrogen is attached with acetyl group with second carbon of the glucose. That's why it is called N-acetyl glucose amine, because it is attached with the glucose with N group. So you can see it is on carbon number 2. When this molecule, the other molecule of glucose will be flipped, so this nitrogen, this N-acetyl glucose amine or N-acetyl will group will be flipped also, because it is attached with carbon number 2. So here carbon number 1 and carbon number 2 will come here. So like that, the alternate molecule is flipped upward or downward, upward or downward. So this make the same arrangement as it is in cellulose. So actually this makes a beta 1,4 glycosidic linkages, and that is the function is the same as it is in the cellulose, that it is the structural carbohydrate polysaccharide, monopolysaccharide, but it is present specially or mainly in fungi and exoskeleton of arthropods. So dear students, up to here we have completed our topic carbohydrates, in that we have seen all three types: monosaccharides, oligosaccharides, and polysaccharides. And in polysaccharides we have seen monopolysaccharides and their structural formulas and how they make the linkages as well. Inshallah, in the next lecture we will continue the next topic. Till then, Allah Hafiz, wish you all the best.